Okay, so good afternoon and welcome to the Biochemistry and Molecular Biology Departmental Seminar Series. It really is my pleasure to introduce our speaker for today, a colleague and an associate professor in the Department of Biochemistry, Dr. Malcolm Burns. Before I hand over to Dr. Burns, I would like to give a short bio of his accomplishments. So Dr. Burns earned his PhD degree in biochemistry from the Louisiana State University in Baton Rouge. He was an NIH postdoctoral fellow in molecular medicine at Cornell University in New York, and subsequently taught various subdisciplines of chemistry, including biochemistry, at several universities or colleges, namely Xavier University, the University of Louisiana at Lafayette, and St. John's College in Belize City. He joined Howard University in 2001 after spending 2 years as a research scientist in the biotechnology division at the National Institute of Standards and Technology in Gaithersburg, Maryland. While at Howard University, Dr. Burns' research focus has primarily been on structure and function of enzymes of potential biomedical and biotechnological importance for bacteria and archaea. Please indulge me a little while I give some background to this seminal article that will be presented shortly. In 2004, while reading about the new field of ecological developmental biology, which is a subdiscipline of evolutionary developmental biology, Dr. Burns began to notice the pioneering work of a biologist named Ernest Everett Just, and has written a number of papers and given several talks on the work of Ernest Just. In 2008, Dr. Burns teamed up with Stuart Newman, whose paper is the topic of today's discussion. And Dr. Burns co-organized an NSF-funded symposium on Ernest Just's work with no other but Dr. Stuart Newman. Drs. Burns and Newman have co-authored a paper entitled Ernest Young Just, Egg and Embryo as Excitable Systems. This was published in the Journal of Experimental Zoology in 2012. And they have both been invited to speak, and they were both invited to speak at the International Symposium on Just at the Marine Zoological Station in Naples, Italy. So I will welcome Dr. Burns to give his seminar, which is entitled Key Features of the Metazoan Gene Regulatory Ground State, a journal club style discussion of Newman A. Stewart article, which was published in 2020, known as Cell Differentiation. What have we learned in 50 years? Thank you, Dr. Burns.
Thank you, Dr. Quabiado. It's a pleasure. It's a pleasure to speak to you guys and to give a presentation to the department. I'm going to share my screen now. So once again, thank you. And what I'd like to talk about today are some of the key features of the Metazoan gene regulatory ground state. A couple of years ago, I read this article by Stuart titled Cell Differentiation, What Have We Learned in 50 Years, published in the Journal of Theoretical Biology in 2020. And I was amazed. I was amazed by, the recent discoveries in the area of regulation of gene expression in higher eukaryotes, the metazoa in particular, and the fact that liquid-liquid phase separation undergirded a lot of that, which Stuart uses to say that the usual models we have of explaining gene regulation in metazoa are not adequate, no longer adequate. Here's the main thesis of Stuart's paper. And this is a quote from the article. cell types, rather than arising by adaptive selection and then diversifying within a species by modification with descent, are actually emergent physiological manifestations of inherent cell functions. Such functions could have been mobilized essentially automatically by the novel gene regulatory system that appeared coincidentally with Metazoan multicellularity. And I want to tell you a little bit about Stuart. Bernard mentioned that, we teamed up together to co-organize a symposium in 2008 on Ernest Everett Just. We co-authored a paper together later on, and we also were both speakers at a symposium honoring Just in 2012 in Naples, Italy. But I've also followed, so we have a mutual interest in Ernest Everett Just, but I've also followed his work because I find it to be very fascinating. He works in the area of cell and developmental and evolutionary biology. And a lot of his ideas are pretty out of the box and kind of novel ideas about how cells and organisms arose in evolution. So he's a professor of cell biology and anatomy at the New York Medical College in Valhalla, New York. His research areas include the role of what's called developmental patterning modules, which is a term that he coined, in the origination, development, and evolution of complex organisms, epigenetic mechanisms in the origination of organismal forms, and generic physical mechanisms of morphogenesis and pattern formation, the origins of multicellularity, embryonic limb development, inherency and agency in the origin and evolution of biological functions. And I want to define inherency here because it's a critically important term for Stuart. It's the idea that aspects of the phenotype are latent in an organism's material identity and will emerge spontaneously under the right conditions. Certain structural motifs, for example, tissue layers, lumens, segments, appendages, are readily generated by physical organizing forces acting on tissue masses with minimal programming from the genome. So what happens during evolution is that forces that are latent in the organism are mobilized through innovation, allowing for evolutionary change. And so what you don't have is the gradual gene-driven type of change that you see with the modern synthesis, the modern evolutionary synthesis. He's authored 2 books, more than 2 books, but he's co-authored these 2. Biological Physics of the Developing Embryo, published in 2005 with Gabor Forgax, and Biotech Juggernaut, Hope, Hype, and Hidden Agendas of Entrepreneurial Bioscience, published in 2019 with Tina Stevens. So Stuart also has is concerned with social issues surrounding heritable gene genome editing and genome editing of human embryos. So he has that sort of social and ethical concern, and he's written about that. He's a very broad thinker. He's also the editor-in-chief of Biological Theory, a journal. At any point, if anyone wants to stop me and ask a question, I want you to feel free to do that. Okay. I might just, so I just want to say that. All right, so let's keep going. So in this paper that Stuart wrote, the topic is, the title is, what have we learned in 50 years regarding cell differentiation? And in 1969, that's about 50 years ago, 2 models were proposed for how genes are regulated in higher eukaryotes or higher eukaryotic cells. One was called the Global Genome Regulatory Dynamics, or GGRD model, proposed by Stuart Kaufman. And the other was called the Modular Gene Regulatory Network, or MGRN model, of Roy Britton and Eric Davidson. These two models have changed over time since 1969, and today both use a Boolean true-false, either-or, yes-no type of logic, and rely on the concept of a genetic switch derived from bacterial gene regulatory processes, such as the lac operon, that were known at the time. So this was in 1969. By the way, please ignore the hammering, which you may hear in the distance. I apologize for that. You might also hear a bird every now and then. So these models were derived from bacterial gene expression mechanisms, which makes sense because that's what was known back then. But so those were the two main models back then. And in the ensuing decades, aspects of these two were combined, the strengths of each, and a consensus, a dynamic plus modular model, was proposed or has been proposed. And this is now the default model that's used to construct virtually all developmental simulations. So I want to give a couple of examples of applications of some sort of consensus of these two models. Now, this is a diagram, a figure. It's a figure 3 from the paper that was actually written about 20 years ago, in which Eric Davidson was the first author. Another author on the paper was Leroy Hood, who is a very innovative scientist who actually invented a type of what's called gene sequencing. It's a recent type of gene sequencing that was picked up by no, this is skipping me. Applied Biosystems. He actually started the company Applied Biosystems, and automated DNA sequencing was a technique that he invented. So he's been very important in the area of genomics and proteomics and systems biology, and he actually founded the Institute of Systems Biology at University of Washington. This is Leroy Hood, who is a co-author on this paper. So it was really a blockbuster paper that combined the efforts and the expertise of different people. And what you can see, and I'm not going to really unpack this, I included the figure in case you want to look at it later, but it's very detailed, sort of grid-like, switchboard-like circuit board pattern of transcription factor action in stimulating, in activating and repressing different downstream genes through the developmental process as endoderm is converted to mesoderm in the sea urchin embryo. And it's really quite amazing. And when I first read this back in 2002, I thought, wow, this is really amazing here, okay? And people could do that then because you could do proteomics and figure out which transcription factors were present. You could do interactomics to see what's interacting with what. So it's a very exciting thing. And here's a quote, though, from the paper. This model represents an outline of the heritable developmental program. The organismal systems that constitute the machine sum to the majority of the working parts of the cell. Their mobilization is controlled by the transcriptional switches that hook them into the genomic regulatory control system. So this language is actually something very familiar to us, right? The idea that they're transcriptional switches, right? Going back to the original idea of a genetic switch. So note how reductionistic this language is. It's talking about the cell as a machine. So I'll turn my attention now to one more example of an application of these models, these early models that have, developed over time and been revised and refined. This is one looking at the reconstruction of the transcription factor regulatory network for the reprogramming of mouse embryonic fibroblasts to induce pluripotent stem cells, or iPS cells. And it takes data from experiments done using the 4 Yamanaka factors so that the MEFs were reprogrammed into iPS cells over a period of days, maybe a week or more. Okay. So they reprogrammed those mouse embryonic fibroblasts using the Yamanaka factors. And in this paper, Choli et al. Created a program they called Tetramer, which stands for Temporal Transcription Regulation Modeler, to combine data from multiple gene regulatory network sources and reconstruct a transcription regulatory landscape, which we see in figure D here. By the way, this acronym is torturous, and it rivals the acronyms of the NIH. I'll just say that. What they did was they looked at which genes are being expressed with transcription factor genes over 8 days, right? And then look to see what interacts with what, and which of the transcription factors were what they called called the master regulatory index. Which ones were master regulatory index transcription factors, sort of the master transcription factors. And they picked the and they picked a set of them and then looked at how they all interact and over time, and came up with this network here, which is another type of manifestation of this sort of an interactome here. One thing you can notice is that after about 5 days here, you begin to see this is OCT4, NANOG, SOX2 being expressed endogenously. So the Yamanaka factors then trigger or stimulate the endogenous expression so that you have self-renewal in these cells. So, this kind of work is useful to a point and does reveal some information. But what Stuart is saying is that it doesn't capture what's really taking place. And so what I want to do now is talk about what does take place. So here are the problems with these 2 original models and those that are derived from them. So we now know that gene regulatory systems in metazoa, which are the animals, are quite different from those in bacteria. And there are 2 main facts that undermine these classical switching network models. First, most transcription factors, about 90% in animals, contain intrinsically disordered protein regions, or IDRs, which introduce unpredictability in binding and interaction patterns. Second, transcription factors and other IDR-containing proteins and cis regulatory enhancers exhibit their variable effects through the formation of phase-separated liquid condensates, which control transcription. So you have a totally different paradigm here, not a switchboard, but this is happening through these liquid-liquid phase separation type condensates. So this switchboard-like circuit diagram is just no longer adequate, okay? So we need a new model. Stuart's not proposing a new model, really, because it's still early days and there's a lot we don't know. But he's pointing out the inadequacy of these earlier models. So this is a bit of a busy slide, actually, and I'll try to move through it quickly, about intrinsically disordered proteins and protein regions. This phenomenon was not recognized until the mid-1990s. IDPs fold into stable defined structures. They fail to fold into stable defined structures as free monomers, but they can become ordered when they bind to a protein partner. In the absence of the binding partner, they're dynamically disordered and fluctuate through a range of conformations. They're characterized by a biased amino acid composition with few hydrophobic amino acids and higher than usual numbers of charged and hydrophilic ones. We'll take a look at that later. They can act as hubs in protein interaction networks, integrating multiple signaling pathways and acting as scaffolds that bind proteins from different pathways. So they're important in the regulation of transcription, and that's what I'm going to talk about here, and in translation and in the cell cycle as well. By coming together, they promote phase separation in the cytosol and in the nucleus, actually, to form membraneless organelles that serve to compartmentalize cell components. They're sites for post-translational modification, such as phosphorylation, and they are encoded in segments that are involved in alternative splicing. So these three, IDPs and IDRs, post-translational modifications, and alternative splicing function synergistically together to regulate cell function. And here are some examples. We can look at the disordered tails of histone H1 proteins that collect combination of post-translational modifications and constitute what's called the histone code that regulates gene expression. The disordered kinase-inducible domain of CREB binds and adopts a helical conformation on the KIX domain of the CREB binding protein, triggering transcriptional activation of downstream response genes. And I'll talk more about that in particular. And then finally, the tumor suppressor protein P53 has a disordered C-terminal domain that can fold into various shapes as it interacts with different partners. So it displays conformational promiscuity as it controls cell division. So this is sort of what this is all about, intrinsically disordered proteins and protein regions now, sort of as an introduction. So getting back to the paper now that Stuart wrote, he wrote about some of the components of the Metazoan gene regulatory ground state. And some of these components actually pre-existed in ancestor pre-Metazoan cells and were mobilized in a different way upon a transition to multicellularity or further evolutionary development. Others were novel innovations that occurred when Metazoa arose through evolution. An example is enhancers, which are they're Metazoan specific hallmark of the Metazoa. So I'm going to talk a bit about enhancers and something called super enhancers. These are cis regulatory elements on the DNA. Pioneer transcription factors, which actually bind to cis elements on the DNA and help to open up, sort of pry open the chromatin there, allowing other factors, these lineage determining transcription factors, to then bind and associate. I'm going to talk about the CREB binding protein and P300, which is an histone acetylase and transcriptional coactivator. Mediator, which acts as a molecular bridge between transcription factors and polymerase II. Something called topologically associating domains, or TADs. Liquid-liquid phase-separated transcriptional condensates, or hubs. I'm not going to talk about gene regulatory silencers, and that's why I have a little more here on this slide. These are also cis regulatory elements like enhancers, but they're the repressive counterparts of enhancers. They're associated with polycomb group proteins, PGCs, and other lysine methyltransferases such as SUV39H, which act by methylating histone H3, thereby moving the promoters that they methylate, moving the promoters that are associated with those histones into an inactive heterochromatin state, thereby silencing them, okay? And the silencing occurs through phase separation of silencing complexes into a liquid-like condensate. So they have that in common with enhancers, as we'll see shortly. What's also interesting is that enhancer-derived RNAs called eRNAs also are part of this this silencing process. And then also talk about the polymerase II pre-initiation complex. So I'll talk a bit about enhancers and super enhancers. So again, going down to the last bullet here, enhancers are hallmarks of Metazoan gene control. So they're absent from extant or existing unicellular holozoans, and they're sort of a hallmark of Metazoan gene control in particular. So these are cis regulatory elements, about 100 to 1,000 base pairs in length, that are typically located far from a gene and its promoter, they can be upstream, downstream, within an intron. They contain dense clusters of transcription factor binding sites. They bind cell type specific transcription factors, which then recruit histone deacetylases such as CBP300 via their activation domains that contain intrinsically disordered regions. Okay, so they recruit these histone deacetylases. Once the chromatin then opens up because of the acetylation, a large protein called mediator associates and acts as a molecular bridge between the transcription factor activation domains on the from the enhancer and polymerase II. Okay, so it recruits mediator, and the mediator can act as that bridge with Pol2. So enhancers contact promoters directly through formation of what's called of chromatin loops and topologically associating domains, TADs, that contain, and these condensates that form contain transcription factors, mediator, and the Pol2 initiation complex, among other proteins. And I'll talk about those large condensates and what they contain and how it works, how they activate transcription. So bursts of transcription from enhancers can occur, and they generate these enhancer-derived RNAs, eRNAs, which may play roles in activating transcription and also in silencing, as we just saw. Enhancers have highly cell type specific histone modification patterns, and they strongly correlate with cell type specific gene expression patterns. And there are around 150,000 enhancers in the human genome. So there are many more enhancers than there are genes. By the way, when I was reading that paper, the original paper from 1969 by Stuart Newman, one thing he said in there, he predicted there would be 2 million human genes. Well, back in 1969, that may have been, not too unreasonable, but now we know, of course, there are only around 20,000 genes in the human genome. So that's one thing about that paper that wasn't quite right, but that's okay. It was 1969. So what are super enhancers? These are clusters of enhancers that mediate cell differentiation in association with these LDTFs, such as MyoD, activin muscle, and RUNX2, activin bone differentiation. And I mentioned the pioneer transcription factors. These include OCT4, FOXA, SOX2, GATA-A4, and these are the first to bind, and they help the chromatin to open up, allowing other protein factors to join at the site. By the way, OCT4 and SOX2 are two of the so-called Yamanaka factors that stimulate pluripotency. All right, so that's enhancers. Again, they're metazoan specific. And now, not so many words now, not so much text. Let's take a look at the Krebs binding protein, or CBP, and its paralog P300. This is an amazing protein, absolutely amazing. Here's a sort of a diagram of its quote-unquote structure, and here's a schematic diagram of the domains that it contains. One thing you'll notice is it's a multi-domain protein, and also you'll notice that not all of the domains are represented in the structure shown in part B. And that's because those domains are intrinsically disordered. Other domains are intrinsically disordered in the free state, but then when they bind a partner, adopt a structure. And an example, and this domain called NCBD actually exists in a molten globule state that then folds into a structure when it binds its partner. And we could look at a few out of these, the histone acetyltransferase activity is part of this gray domain. This is a histone acetyltransferase protein. And so TAS 1 and 2 are zinc binding domains that it's called the transcriptional adapter zinc binding domain 1 and 2, and they all have different interaction partners. And so shown on the right is interactions of CBP P300 domains with arrays of different transcription factors that are assembled at promoters for the transcription of different genes. So you see how CBP then can interact with different transcription partners through its different domains, and the disordered regions between those domains allows for great variability in what they interact with. It's just an amazing, and so it's an amazing protein. So once they bind in to particular transcription factors upstream of a gene, they acetylate the histone, and that leads eventually to activation of transcription. So this is CBP. It has a lot of intrinsic disorder. And it's part of these condensates that form. So let's keep going. Now, mediator, it's that bridge, that molecular bridge, right, between the enhancer and PAL2. And so here we see on the left a structure of mediator. It's a huge protein. It's about 1.4 megadaltons in size, and it has 26 subunits, some of which are highly disordered. And that's shown here in this, what's a top-down view of the structure of mediator. And you can see these shaded regions here, the green shaded portions of MED14, MED25, MED15, MED1, and MED26. The dotted ones are in the back of this structure, and the solid ones circled by the solid lines are in front. But look at MED1 there, just completely disordered. So what does that tell us? One, it's going to be able to associate with other proteins within a liquid condensate, and it can bind different partners. So what do we see here? We see how mediator, shown in green, bridges between the transcription factors bind to an enhancer, okay, and a PAL2 initiation complex here. We can see some of the parts of PAL2 initiation complex, such as transcription factor 2H, this kinase domain. Here's the promoter from which expression is being stimulated. Now, this large chromosomal loop and the associated condensate is called a TAD, a topologically associating domain. You can see PAL2 molecules becoming a part of this initiation complex, and you can see, what's going on is you can see the transcription occurring with the mRNA transcript being produced. And then what this shows also is how transcription bursting can occur. So it's showing how as long as there's a transcription factor still bound to mediator, it can reinitiate another round of transcription. And that saves time and allows for this rapid bursting, so that, because if it had to find this enhancer, again and again, that would not allow for this kind of rapid bursting to occur. The complex is already formed here, as long as the transcription factor remains bound. I do want to say that cohesin and CTCF are responsible for forming this TAD, this large chromosomal loop that makes up part of the TAD. Those are two important proteins there. So let's keep going. So I want to talk about these liquid-liquid phase-separated transcriptional condensates, or hubs. Here's a paper from 2018 by Boija et al. Titled "Transcription Factors Activate Genes Through the Phase Separation Capacity of Their Activation Domains." One thing I failed to emphasize was that transcription factors typically have two domains: a DNA binding domain and an activation domain that can interact with other protein partners. And it's that activation domain that has the intrinsic intrinsically disordered regions. That allows them to, interact with partners and also be a part of these condensates. So what these authors found was that transcription factors form phase-separated condensates with mediator. Phase separation of activation domains is a general property of transcription factors. Phase separation capacity of transcription factors is associated with gene activation, and these condensates containing transcription factors incorporate dynamic and structured interactions. And so we see here a diagram showing the condensate in blue shading. DNA, what do we have there? An enhancer shown with the orange bar, and then transcription factors bound with their disordered activation domains, now interacting with co-activators that also have intrinsically disordered domains, such as CBP. That's an important co-activator. And these are all interacting here in this large phase-separated liquid droplet in the nucleus of a cell. There are different types of interactions you can have here. Interaction among the intrinsically disordered regions involving different types of residues, and more structured interactions. There also can be present. There. So moving along, talking about another component is the mammalian pre-initiation complex. Now, one thing I want to say here is that some of these structures are present in lower eukaryotes. And so they were sort of mobilized in the process of multicellularity, of the rise of multicellularity. They're not unique to Metazoa, of course, we know that. Anybody who teaches, regulation of gene expression and transcription knows that you have a pre-initiation complex that looks a lot like this in yeast, for example. But this structure was of the mammalian human pre-initiation complex was solved in 2021. And the reason why it took so long, from what I can understand, is that there are some regions of PAL2, of the complex rather, that are disordered. And so they pose a challenge in determining the structure. So this complex is highly dynamic, especially owing to the disorder within transcription factor 2H, TF2D, which is not shown, and mediator, which interacts with it. So I don't know why TF2D is not shown here. It's a general transcription factor that's the first to bind to the promoter and upstream of the promoter and initiate transcription. So I'm not sure why it's not here. But maybe you just couldn't, I don't know why it's not part of the structure. But this is the pre-initiation complex, multi-component. You can see the DNA here. It's bound to the DNA. This is PAL2, and these are all these transcription factors with their some subdomains and indicated, like the TATA binding protein portion here. So on the right, we have the pre-initiation complex shown at the bottom. You can recognize its structure. And on top is mediator. And this is what? Is this a side view? I think this is a side view. And down here we see a top view, which you see mediator up on top like this of the structure. So this is how mediator interacts with the pre-initiation complex. Bringing the enhancer together with the promoter and stimulating and activating transcription. Is that all I want to say there? One thing I want to say is that when PAL2 has a cytoplasmic tail that, when phosphorylated, causes elongation, transcriptional elongation to proceed. So that's a very important event in the activation of transcription, and I'm going to talk more about that in just a minute. So how did cell differentiation in animals evolve? Right? What is Stuart Newman saying in this paper? He's saying that the Metazoan specific gene regulatory apparatus is based on, quote, fluid state physical and mechanochemical effects occurring at condensed hubs that are sites of topologically plastic interactions among molecules with conditional identities. So how did ancestral pre-Metazoan cells evolve into the different cell types of Metazoans? He asked this question: were ancestral cells preloaded with sets of genes that had modular functions that then took on new roles with the advent of enhancers and a broadened functionality accompanying the rise of Metazoa? And that's the question he's asking. This view, which relies on the mobilization of pre-existing inherent functions, runs counter, as I mentioned earlier, to the gradualism of and gene-driven adaptive change that's assumed by the modern synthesis. So that's sort of his thinking about it, that once you have this happening with formation of these with this liquid-liquid phase separation and these physical forces driving the activation of transcription, it's not as simple as we thought. Okay. I want to sort of begin to close out with two recent studies that were published since Stuart wrote his paper. This is actually a few months later, in 2020, by Wei et al. Out of Cliff Brankwin's group at Princeton. It's titled "Nucleated Transcriptional Condensates Amplify Gene Expression." And this figure to the left here is a model that they present for the role of condensate nucleation in causing transcriptional bursts. Okay, and so we see what's going on here. Here's PAL2 associated with a promoter of a gene. There's no transcription occurring, but then, they dealt in particular, by the way, with a set of proteins, a family of proteins known as FET, which involve FUS, EWS, and TAF915. These are transcriptional regulators, and they all contain N-terminal intrinsically disordered regions. These proteins also are involved in DNA repair and also in pre-mRNA splicing in the nucleus. So he looked at those proteins in particular, and he saw that they form these condensates, okay, which grow in size as additional C-terminal domain recruitment occurs. So PAL2, via its C-terminal domain, can recruit other factors that arrive and help form this condensate. Once it forms, it activates transcription, and you can see. But then what happens, as I mentioned, is that C-terminal tail becomes phosphorylated. That's what allows transcription to proceed through elongation, and it also causes the release of PAL2 and the destabilization of the condensate. Right, so the condensate can wax and wane, can grow larger and smaller. It's a very dynamic structure. And in the middle here, I just show this technique, or this approach that they developed in the Brangwin lab. It's an optogenetic approach. And it's really cool. Basically, they took part of what's called the cryptochrome 2 gene of Arabidopsis thaliana, which can dimerize or oligomerize in the presence of blue light. And here you see it, the orange Cry2 domain, and used that and attached it to intrinsically disordered regions from proteins, from these FET family proteins like FUS and others, and DDX here is a control, by the way. but they attach these intrinsically disordered regions here, and then express those in cells, and notice that when they expose the cells to blue light, these structures formed. Okay, and these were the condensates that were forming, triggered by the light. So they had a nifty optogenetic approach they could use to study the formation of these condensates in live cells. That was an impediment. So they developed a new technique that now can be used. And then finally, the second recent study was just published this year. It's by Lyons et al., and this is through out of the Sabari group, Benjamin Sabari at UT Southwestern. And the title of that paper is Functional Partitioning of Transcriptional Regulators by Pattern Charge Blocks. So here's what they found. Condensates composed of IDRs of MED1, that's one of the subunits of mediator, selectively partition RNA Pol2 together with its positive allosteric regulators. Negative regulators are excluded. So it's a rather specific way of incorporating activators, which is kind of amazing to me, since it's a physical process. The partitioning compartmentalization is sufficient to drive gene activation during cell state transition. And let's look at the figure that shows that. So here we have, selective partitioning of the positive regulators of RNA polymerase 2 with Pol2 itself. See, they're forming this condensate, okay? And then it's the charge pattern in the disordered regions that drives selective partitioning. So there's a particular pattern of positively and negatively charged residues that when you remove that pattern, it no longer partitions. You see how these do and those don't? Meanwhile, you can have an IDR that lacks the particular pattern of charged residues, but when you modify it, add that pattern into it, it will partition. So it's the pattern of charged residues in that IDR that's allowing it to partition into this condensate. And that drives, they showed, gene activation.
Dr. Burns, is 2:55.
All right, so I'm almost done. So Dr. Newman, at the end of his article, quotes E.E. Just. He says, while it may eventually turn out that cell types are the behavioral modes of a single physical system, as suggested by the GGRD model, it will be a system embodying phase transformations of protein nucleic acid complexes, the principles of which defy familiar laws. So he's saying, whatever model we propose, it's got to embody phase transformations, because that's really important in metazoan control of gene expression. And then he says, in the words of the mid-20th century biologist E.E. Just, written 30 years before these models were proposed, understanding development will require, quote, of physics and chemistry in a new dimension, superimposed upon the now known physics and chemistry. This was from Just's book, The Biology of the Cell Surface, published in 1939. So finally, he concludes by saying, we will see what the next decades bring. Here are the sources I consulted, and I'm ready for questions if you have any.